Okay, so now the second part of our lecture 4 is we're going to discuss the digestion of carbohydrates. And this, of course, is all surrounding glucose metabolism, so nothing different. So this is just a very big overview, and don't worry, we're going to go through all of these different reactions in more details as we go through this part of the lecture. So again, we're starting with the intake of starch, which is basically glucose molecules put together, lactose, sucrose, sugars that are going to have to be broken down into simpler sugars. We have our alpha amylases. There are two locations where you find these alpha amylases. They're exactly the same. There's no difference. It's just that one starts in the mouth. So you have your saliva alpha amylases. And as they begin to break down your sugars, as they pass through the stomach, they get inactivated. And so your pancreas also can secrete alpha amylases. We'll pick up on the sugars where the saliva alpha amylases left off. So the saliva is going to start in the mouth, and then the pancreas will release the enzyme to continue in the intestine. Alpha amylases are inactivated by hydrogen chloride, which is our stomach acid, and so that's why they are made both in the mouth and in the pancreas. Now we have on our brush border our disaccharidases. So as these sugars get broken down into twos, then the brush border have a series of disaccharidases that can break down those twos into single sugars that can then be absorbed. Remember, we have our GLUT receptor, a GLUT5 receptor, as well as our sodium transporter, but they can only transport in monosaccharides. They cannot transport in disaccharides. So on the brush border, your disaccharides will break those down into monosaccharides. So we have glucoamylase, which is going to hydrolyze alpha-1,4 bonds of dextrins. We'll talk about dextrins in a moment. We have our Sucrase-Isomaltase Complex, and that's going to hydrolyze sucrose and most of the maltose, isomaltose from our glucoamylase reactions. We have a Lactase Glucosylceramidase, which is also referred to as B-glycosidase, which is going to hydrolyze beta-glycosylic bonds in lactase and glycolipids. So over in this picture, it shows you are eating the sugars. You have your salivary alpha amylase. In the stomach, you get your sucrose and lactose that get passed on. You then have your alpha dextrins that are going to pass into your small intestine. Your pancreas will release your alpha amylase again. And in the small intestine, you'll have tri and disaccharides that are going to be worked on by the different types of enzymes. And then you're going to have your maltase, isomaltase, sucrose, lactose that will have your brush border enzymes then make them into the monosaccharides of glucose, fructose, and galactose that can all be absorbed. And just showing down here, fiber gets moved on into the colon, and we'll discuss that as well. Okay, so dietary fiber cannot be digested by humans. We don't have the enzymes for that. And so they're going to be passed into the colon where they're going to be worked on by bacteria and converted to gases, methane, for example, carbon dioxide, and hydrogen. They're also going to become acetic, propanoic, and butyric acids, and these are going to be absorbed and used as energy by your colon epithelial cells. Glucose, galactose, and fructose are going to be absorbed by the epithelial cells, and we have the sodium-dependent active transporter as well as the facilitated diffusion. We have the GLUT5 receptor, which is the fructose receptor. Glucose can also use. Galactose and glucose can both use the sodium co-transporter as well. These sugars are then going to go into the blood, then the liver, because the liver is the first place that you dump whatever comes into the gut. And then the peripheral tissues will take up using those different GLUT receptors on their surface. The facilitated transport of glucose is by those GLUT receptors 1 through 5. And then the type of GLUT receptor expressed is going to influence the metabolism of glucose during the fed and fasting stage by those different cells. All right, so let's talk about carbohydrate digestion. Of course, 40% of our calories are going to be carbohydrates, usually. They're going to be digested by specific enzymes. We have starch, which comes from plants. We have the alpha amylases that's going to work on the starch molecules. And then we also have lactose, which is going to be worked on by beta-glycosidase, lactase activity. We have sucrose, which is going to be worked on by sucrase. Fructose, from high fructose syrup, we can absorb fructose directly. And then we have cellulose, which is a fiber that we can't break down. So remember, our lactose is galactose and glucose. Our sucrose is glucose and fructose. And you can see these bonds. So between galactose and glucose, we have a beta 1,4 bond. Between glucose and fructose, we have an alpha 1,2 bond. And then remember, our branches are always going to be alpha 1,6. So this would be like in our starches. We have an amylose, which is the alpha 1,4 bond. We have an amylopeptin, which is a series of amylose. We have these alpha 1,4s. But we also, on our amylopeptin, we are joined by the alpha 1,6 bond as well. So basically, the enzymes that are breaking these down are going to be specific for these bonds, and they're going to be specific for these bonds either in the middle of a molecule or at the ends of a molecule. So basically, glucose, we have these monomers, and we have starch, which is a polymer of glucose with these branches, similar to our glycogen stores. They are going to be broken down by pancreatic and salivary amylase. They're going to be turned into what we call limit dextrins, which you can see are just smaller versions of those. We also have disaccharides, such as maltose, that gets formed. These disaccharides will go to the brush border. They'll be worked on by brush border disaccharidases, and they will be cleaved into monosaccharides. And the monosaccharides are going to enter the epithelial cells, diffuse through into the blood, and then be transported. So digestive processes are to take all of these complex sugar molecules and break them down into monosaccharides. And this is done by hydrolyzing glycosidic bonds between the sugars. Different enzymes are going to have different specificities with the different sugars. Lactose and sucrose are disaccharides, again, beta 1,4 and an alpha 1,2 glycosidic bonds. This picture over here didn't come over very well, but I think we can still work through this. Okay, so we have starch, lactose, sucrose, which are important in our diets. Starches are going to be amylose and amylopectin. Again, the amylose is a smaller without the branching, has the alpha 1,4, and the peptin, the amylopeptin, are going to have the branches. So they're going to be the dextrins and the limit dextrins with the alpha 1,6 and the alpha 1,4s. I'm showing here amylopectin. See what alpha 1,6s, and these are alpha 1,4s. And this can be larger, of course. Digestion of the starch is going to begin in the mouth again. The alpha amylase is an endoglucosidase. It hydrolyzes internal alpha 1,4 bonds between glycosyl residues, and it does this randomly, so it's going to make random-sized dextrins. Salivary alpha amylase is going to be inactivated by our stomach again. Your pancreas will release bicarbonate, which will neutralize the pH, and also pancreatic alpha amylase. And this will continue to digest those amylose and amylopectin molecules. Now, important, alpha amylase cannot work on those branches. So there has to have another enzyme that's going to have to work on those alpha 1,6 branches. So this is the action of salivary and pancreatic alpha amylases. Again, they're going to attack those alpha 1,4s randomly, internally. They don't work on the reducing and non-reducing ends. They only work on the middle. And then you're going to have different sizes that result. You'll have a disaccharide maltose. This can go to the brush border and be broken down into glucose and then be absorbed. You also have isomaltose, which will have that 1,6 bond that alpha amylase cannot work on, and so we'll have to bring in another enzyme to work on that. You also could have a trisaccharide, and so this is larger than the maltose, and this again will have to continue to be digested until it gets to the disaccharides, so the brush border enzymes can work on them. And then we have our alpha dextrins. Alpha dextrins are going to be longer with the alpha 1,6 and a little bit bigger than our limit dextrins, which is the example there is isomaltose. So eventually, with respect to your alpha amylase, you're going to have residues with alpha 1,6 bonds, and the alpha amylase cannot work on those. It has only activity towards alpha 1,4 bonds. Alpha amylase also has no activity on alpha 1,4 bonds at the non-reducing end of a chain. So just keep in mind what we mean by the the non-reducing end. So the non-reducing end, let me do it this way. So let's say reducing end of a sugar molecule is where you have a free carbon number 1. So that's that anomeric carbon, and that's the reducing end. And then the non-reducing end is all other ends. So for example, I'm not going to draw the whole structure, but here's glucose, here's carbon 1, and that's the end, and so this other carbon over here would be a non-reducing. And then if there was a branch up here, this would be a non-reducing. And this one is the reduce end. Okay, everything else is non-reducing. Okay, so in the small intestine again, our disaccharidases are going to finish the job. We're going to have a series of other enzymes that are going to work on different sizes of the dextrins and limit dextrins to hydrolyze the alpha 1,6, the different 1,4s at the non-reducing ends, etc. So let's talk just a little bit about our disaccharidases. These are going to be on the intestinal brush border. The disaccharides are going to be converted to monosaccharides by these, and so monosaccharides, of course, get absorbed. Glycosidase activity is going to be in 4 different glycoproteins. We're going to have glucoamylase, a Sucrase-Maltase complex. We're going to have a trihalase complex, a Lactase Glucosylceramidase complex as well. So this is a table from your book that kind of describes all the different complexes. We have a beta-glucoamylase. This is going to split the alpha 1,4 bonds between glycosyl units, and this is going to be starting at the non-reducing end of the chain. So again, our alpha amylases could not work on the non-reducing end. Our beta-glucoamylase can, which the catalytic site is our alpha-glucosidase. It's called an exoglycosidase, and it's going to work on those amylose and the amylopectin that were made by the alpha amylases, as well as glycogen and maltose. It also has another site. It's a beta-glucosidase that can work on the same things that the alpha-glucosidase did, but it has just different specificities. So, but together they can break down a lot of the molecules. You also have a Sucrase-Isomaltase that has 2 different sites, a sucrase-maltase, and this is important for splitting the sucrose molecule, because remember sucrose has the alpha 1,2 bond. So sucrase-maltase is going to be important for being able to metabolize basically all the sucrose. It also can work on the maltose and the maltotriose. The other side is the isomaltase-maltase, which splits the alpha 1,6 bonds in those limit dextrins that we talked about, those branching points. And so that's very important. And it can also work on the alpha 1,4 bonds. The beta glycosidase is going to have 2 sites as well. It's a Glucosylceramidase, which is going to break the beta-glycosylic bonds between glucose and galactose and different hydrophobic residues, such as with glycolipids, ceramide, glycosylceramides, etc. It also has the lactase activity, so this is the beta-1,4 between glucose and galactose in lactose. Trihalase is going to split the alpha-1,1 bonds through anomeric carbons that are bound together. This is going to be important in insects, for example. But we don't really eat insects, so we don't really digest using the trihalase that much in the United States. So let's just go over these just a little bit. The glucoamylase is going to be similar to the Sucrase-Isomaltase Complex. They have multiple enzymatic sites, both of these complexes. They have two globular domains that contain the catalytic sites. Again, the glucoamylase is an exoglucosidase, and it has the specificity for alpha-1,4 beginning at the non-reducing end, any of those non-reducing ends. And it's going to take those limit dextrins down to isomaltase. So it's going to work on what the alpha amylases left off and generate the isomaltases. It doesn't work on that 1,6 bond, for example. There is another site on the glucoamylase that can hydrolyze the 1,6 bonds. And then so that will leave it to monosaccharides, and then they can get absorbed. The Sucrase-Isomaltase complex is going to have maltase activity as well as isomaltase activity. It's going to be able to split all the sucrose, maltose, and maltotriose molecules. It's going to split the alpha-1,4 of limit dextrins and of isomaltase. It also has alpha-1,4 sites of maltase and maltotriose. This guy counts for 100% of your ability to hydrolyze sucrose and 80% of the maltase activity. The other activity for maltase is in the glucoamylase complex. And again, maltase, the reason that's important is that you can only absorb monosaccharides, and so you have to be able to split two glucose molecules, which is, of course, maltose. And then also maltotriose, you have to be able to split those alpha-1,4 bonds in maltotriose. This also, of course, hydrolyzes those 1,6 bonds. Trihalase has one catalytic site, again, anomeric carbons, 1,1. This is going to be in insects, fungi, and algae, not major U.S. dietary components. But we do have trihalase activity as well. And then the beta-glycosidase complex has the lactase, as well as working on the glucose that is bound to ceramides, for example. And so this is going to be anchored. So you can see this has inositol, so it's a phosphatidylglycan anchor. And then the lactase will hydrolyze lactose, so that has the beta-1,4 activity. And then the other enzyme cleaves the glycolipids, so they will remove glucose from ceramide, for example. They're also important for hydrolyzing artificial substrates, so they can remove glucose from artificial sugars and things like that, and they can hydrolyze them as well. Now, in our colon, our bacteria are going to metabolize a lot of our sugars. So when we take in a lot of starches, we have a lot of amylose, or we have less hydrated sugars, such as what's in beans. These become more resistant to digestion in our small intestine and will enter the colon. And the fiber and undigested sugars of the colon will be broken down by bacteria. And the bacteria are going to use them as a fuel source. They're going to release gases, as bacteria do. They're also going to release short-chain fatty acids and lactate. The short-chain fatty acids are going to be acidic, propanoic, and butyric acids. So 2 carbons, 3 carbons, and 4 carbons. And then these are going to be absorbed by the colon epithelial cells and used as energy. And then gases are going to be released through the colon due to the activity of bacteria. Lactose intolerance is going to be associated with pain, nausea, and flatulence after ingestion of lactose. Lactase activity is high for about 1 month after birth, and then it begins to decline. Most of the world's population's level of lactase activity is 10% of what it was as an infant. Adults have hypolactasia, or adult lactase deficiency, in which there is non-persistence of lactase. Secondary lactase deficiency is caused by protein malnutrition or gastroenteritis. You can have malabsorption syndrome, tropical and non-tropical sprue, or excessive alcohol consumption can lead to secondary lactase deficiency. People in certain areas of the world sort of existed with dependency on milk in Western and Northern Europeans, as well as tribes in Saharan Africa, and the level of lactase remained at or slightly below infant levels, and this is lactase persistent phenotype. Adult hypolactasia is the normal condition for most of the world. Congenital lactase deficiency is also out there, and it's an autosomal recessive inherited disease in which you have lactase activity is absent. So these are infants that when you give them milk, they cannot take it, and so they could have congenital lactase deficiency. Now, a portion of the diet is going to be fiber, and this is not going to be able to be digested by our enzymes. This is classified as soluble or insoluble. Insoluble are going to be cellulose, hemicellulose, and linguins, and soluble are going to be pectins, gums, things that are found in foods to make them more palatable, for example. The bacteria, flora are going to metabolize our soluble fibers to gases and short-chain fatty acids, and again, these are going to be absorbed by the colon. The new guidelines suggest that we should have 19 to 38 grams of dietary fiber per day. This is going to be shown to help in certain diseases, including divertical disease. It's been shown to, if you have fiber, to prevent lowering. It's going to help to lower the blood cholesterol associated with slowing rate of absorption of sugar, so it makes you feel full, and preventing high blood glucose levels after a meal. 10% of our total calories are going to come from bacteria digestion of things that don't get digested by our small intestine. All right, so absorption of sugars. So we've talked about absorption of sugars. We have those 3 monosaccharides that can be absorbed. Complex carbs are going to be digested, but not all at the same rate within the intestines. So some carbs are going to lead to a near immediate rise in blood glucose. And so the glycemic index helps to identify those foods that will enter the blood very quickly and increase blood glucose. And so glucose and maltose sugar, when you have table sugar, etc., have the highest index of 142. White bread has a high index of 100, and then low indexes are going to be yogurt, skim milk, that have lower indexes of increasing blood glucose levels immediately. We've talked about absorption, so we have our facilitative glucose transporters. So we have our GLUT5. So here's GLUT5 and GLUT5. Both glucose and fructose can use them. We also have GLUT2 on the serosal side. So these are our GLUT2 receptors. Galactose and glucose can use the sodium co-transporter. So it's 1 sodium plus 1 glucose or galactose. Now, the sodium co-transporter has to be maintained by a sodium-potassium pump, and the sodium-potassium pump is an ATP-driven pump. It's going to maintain the ability of sodium to enter with glucose. So it's going to maintain the concentration, make sure the concentration of sodium is lower inside the cell so it can bring in the sugars. So again, low intercellular sodium maintained by that pump on the serosal side. The transport is going to be from low sodium in the lumen, from low glucose to high in the cell, promoting the secondary active cotransport. Sodium high in the lumen, low in the cell, and so that's all done by that ATP transporter. We talked about our glucose transporters, but let's go over them again. We have our GLUT1 receptors, which are going to be constitutive. They're going to be on the human erythrocyte. They're also part of the blood-brain barrier, blood-renal barrier, placental barrier, testes barrier, etc. We have our GLUT2, which has the KM. That's going to be on the liver and the kidney. Also pancreatic beta cells. It's also on the serosal surface of the intestinal mucosal cells, as we saw. We have our GLUT3, which is in their brain, constitutive. We have our GLUT4, which is adipose skeletal heart muscle, inducible. So this is the big one that is inducible. And this is by insulin. So when you become insulin resistant, this inducible GLUT4, you lose that. And so that's why you have troubles in maintaining blood glucose levels. And of course, GLUT5 is going to be on the intestinal epithelial cells, and it's a fructose transporter. So this is just a note that the sodium cotransporter of glucose is not a member of the GLUT family, so don't get that confused. So galactose is going to be absorbed through the sodium transporter. You also have the glucose facilitative transporters, GLUT2. And so this is why galactose is primarily going to be metabolized in the liver. Fructose can enter through GLUT5. It can leave through GLUT2, so it can also use that GLUT2. And GLUT5 has a higher affinity for fructose than for glucose. So it'll choose fructose over glucose. But glucose has the cotransporter to go through. In most cells, you're going to have glucose be able to come in, especially when insulin is present. You have different KMs for your glucose transporter. Your GLUT2 has a very high KM, and so the liver will only use glucose when energy levels or levels of glucose in the blood are high. Muscle and adipose tissue, again, you have an insulin-dependent glucose transporter that has to be upregulated to the plasma membrane in the presence of insulin. This is just glucose transporters in the blood-brain barrier. So what's interesting, you have to keep in mind that the brain is highly dependent on glucose. However, there are a lot of barriers to getting into the brain. And so this picture just shows that. So you have your neural tissue, your blood-brain barrier, as compared to non-neural tissue. And some things to point out. So the first is that neural tissue have tight junctions. And so these tight junctions don't allow things to pass through. So if you wanted to get glucose into the brain proper, you could not go through the in between cells because of the tight junctions. They also have narrow intracellular space. So it's not like these wide spaces over here. They lack pinocytosis, so they don't take up things and bring it inside of them. They don't sample their environment like other cells do. They also have a continuous basement membrane. So it's continuous, whereas in other areas it's sort of staggered. And so again, things cannot diffuse into the brain. What they do have is glucose transporters on both membranes. So that is shown here. You have your glucose transporter on the outside, at GLUT3, and or GLUT1, and then you also have it on the other side. So basically what these cells do is they take up glucose and they put it through to the other side and release it into the brain proper, or in this case is a cerebral spinal fluid. And so the cells lining the blood-brain barrier don't use all the glucose they take up. They will release it into the areas that are it's hard to get things into, such as the brain. And then finally, the GLUT4 receptor. We've talked about this already, but as you guys know, the insulin binding to the tyrosine kinase receptor is going to upregulate the glucose GLUT4 receptors that have been internalized. So the GLUT4 receptors are inside of the muscle cells or the adipose tissue, and only in the presence of insulin do they go to the surface and take up glucose. So this is only in the fed state. Okay, and so that's the end of our digestion.